This video is to demonstrate how to inoculate the agar plates that are going to be used to perform the Kirby Bauer test. So this includes adding antibiotic discs to the plate after you've added the bacteria. The supplies that you need to complete this experiment include two different bacterial cultures. So this is both Staphylococcus and E. coli. These are broth cultures that should be no more than 10 or so days old. So if you have older cultures, depending upon when you set this up, you may need to take and re-inoculate a fresh broth for your bacteria. And so this would be a broth-to-broth transfer that we've done before. Once you have a grown culture of each of these two that's not older than 10 to 14 days, you're going to need a candle and sterile swabs, and you're going to be putting these bacteria onto triptych soy agar plates. You're going to need 2 of these plates. And then also included in your kit is a pair of forceps that you're going to need. You're going to use these to place the discs onto plates. And then the discs, they should be in kind of a Ziploc bag that came with your kit that should have been stored in the refrigerator. And then, of course, whenever we're doing things, we should have our isopropanol to disinfect our tools. So we're going to go ahead and get started with this procedure. So these are the antibiotic discs I was referring to. So they came in this green kind of package. And then I have 2 plates that I've labeled. Of course, we always label the bottom, never the lids. And so E. coli is one of the 2 bacteria. So here's a turbid culture of E. coli, so it's grown, and again, not too old. And then we need Staphylococcus epidermidis, so that's also been grown. And the sterile swabs, you're going to need 2, so each package has 2 in it, and then those are the forceps. So those are the things you should have handy, and we are going to get started. So I'm just going to be working with one bacteria at a time. So the first one that I have front and center there is E. coli. And so as usual, we're going to use our best aseptic technique. So that includes flaming tubes when we open them. And then you're going to use a sterile swab and get some of the bacterial culture, and then flame the tube and close it. And then you can put the tube down. You're done with the tube. And then what we're going to do is swab the entire surface of this plate. So very tight strokes, side to side. So in this case, I'm going kind of from the top of the plate to the bottom of the plate, covering as much of the plate as I can. And then what I'm going to do is rotate the plate about 90 degrees and then repeat the process. So notice I'm not getting any more bacteria. I'm just taking the bacteria that I've already streaked onto the plate, and I'm just going in a 90-degree direction from that to make sure that I have the bacteria completely covered. And then I rotate the plate again, another 90 degrees or so, and repeat the process. So this is the 3rd time I'm streaking over the plate. Again, I've only dipped into the bacteria once. And then the 4th and final time, I'm going to streak the plate. So this just ensures that the bacteria are completely covering all of the surface of this plate. Okay. So I'm done inoculating that, and then, of course, my used swab goes into my isopropyl alcohol. And what I'm going to do is get out, there's 3 individual packages in here. So 2 of them are antibiotics. So there's, those are the blank control discs, so there's no chemical in those discs. And then there's also tetracycline, so there are 2 tetracycline antibiotic discs. And then this is bacitracin. And so the goal is to put one of each of those discs onto the surface of the plate. And so what it asks you to do is to disinfect your forceps by dipping it in isopropyl alcohol. And then you're going to pass it through the flame so that it, don't hold it in the flame, but you'll catch the isopropanol kind of on fire temporarily, so be careful with this. And that will disinfect the forceps. And then you're going to grab one of the discs from this package, and you're going to place it onto the surface of the agar. And then using the forceps, kind of just gently tap it to make sure it's adhered well to the surface. So after we've done that, we want to disinfect, so dip it into and then you'll flame it. So you can kind of see the isopropanol will burn off, and make sure it completely burns off and give it a second to cool. And then we're going to make sure we put a different disc on. So again, there's 3 different discs that you're gonna do. So 2 have antibiotics kind of infused into them, and 1 is a control disc. So this is the second of the 2 discs that I'm going to add to the plate. So that's the second disc. And again, disinfect your forceps, so dip it in isopropanol, and then burn off the isopropyl alcohol. Let it cool just a second or 2. It doesn't take too long. And then this is the third and final disc that we're going to be placing on it. So this is the control disc. This has no chemical added to it. And so here is the third and final disc. And again, each one, I just make sure they're is adhered well. So this is the final one, so disinfect and then kind of flame off the isopropyl alcohol. So that plate is done and needs to incubate. And so we have another plate that we need to set up. So that was the E. coli. And so we're gonna basically repeat the entire process on different. So this plate has been labeled as Staph epidermidis. So this is going to receive the Staphylococcus bacteria. So again, we're using a broth culture and a sterile swab to get our inoculum. So using aseptic technique, we'll flame the tube, get some inoculum on our sterile swab, and then flame the tube prior to closing it. And now we're going to swab the entire surface of this plate 4 times in total. So you only get the sample of bacteria 1 time, but you swab it. Again, this is from top to bottom. So that would be once. And then once I'm done with that, I'm going to rotate the plate about 90 degrees and then repeat the process. So this is the second time swabbing the surface of the plate. Once I'm done with that, I'll rotate the plate again, roughly 90 degrees or so, and then repeat. This is the third time swabbing the plate. And then once I'm done with the third time, I'll rotate it again, and this will be the fourth and final time. So again, the goal here is to get the bacteria completely covering the surface of this plate. So you do that by swabbing it the 4 times in different directions. And of course, we want to make sure we disinfect our swab we just used. So now we use that isopropyl alcohol and flame our forceps. Let that go for just a second to burn off all the alcohol that's on it. Let it cool a little bit. And we're gonna place the same, there were 2 discs in each package, so we only used 1 for the first plate. And so that second disc in each of the packages is going to be placed onto the surface of this plate. And again, just a gentle tap. You don't want to push too hard. You don't want to push it into the agar. You just want to make sure it's adhered. And then disinfect your forceps between each one. So that's one of the discs. This is the second one. This is obviously tetracycline as the package indicates. So this is one of the discs that has been soaked in a particular antibiotic. And then gently tap it and make sure it's adhered. Disinfect your forceps and pass it through the flame to burn off the isopropyl alcohol. And then the final disc needs to be placed onto the plate. This is the bacitracin antibiotic. And then make sure the forceps are disinfected to make sure there's no bacteria on them. So that completes the second plate being inoculated. Now these need to incubate. And then I'll continue this video of what to do after these plates have incubated. There is a measurement that you perform after the bacteria have grown. So that'll be the next part of this video. The plate should be inverted and incubated for about 48 hours. After that 48 hours, what you want to do is take photos of the plates like we've done with all of our previous cultures to demonstrate that you've completed the activity successfully. And then you're going to measure the zones of inhibition around each disc. So if you stay tuned after the next part of this video is going to explain how that can be done. So once you record the measurements, there's a data table in the assignment sheet where you would record the measurements that you obtain. And this is actually the final lab activity. And so the only thing that you need to keep moving forward are these plates that you're going to incubate for 48 hours. But all the other cultures can be properly disinfected and disposed of. So again, that is done in a bleach solution, a 10% bleach solution, where the culture should be exposed to that bleach solution for 24 hours before you can put the cultures in the trash and then pour the bleach down the drain with plenty of water following. The isopropyl alcohol that's used to disinfect tools needs to have 30 minutes to be effective, so make sure your stuff remains in there for 30 minutes. And of course, we disinfect our surfaces that we work on both before we start working and after we complete working. The next part of this video shows how to measure the zones of inhibition surrounding each of the discs. So you only can do this after you've given the chance for the bacteria to grow. So again, this is about 48 hours. So you should see growth around the entire plate, but an area surrounding the disc may have an area where the growth has been inhibited, and this is what you're going to measure. So on the right is a picture of a Kirby Bauer test that was performed. And so hopefully what you can see is this cream color is the growth of the bacteria. And so this is called confluent growth when it's grown all together in one kind of giant sheet. And so each of these discs has a different antibiotic. And notice around each disc there is a zone where the bacteria is not growing. This is known as the zone of inhibition. And so based on the size of the zone, you can determine if this bacteria, and so the bacteria that was plated on here was a species of Staphylococcus. So is this Staphylococcus sensitive to this particular antibiotic, or is it resistant to it? So that's what you're trying to figure out by performing this Kirby Bauer test. And obviously you can do this for multiple antibiotics all at once because there are different zones of inhibition. So I'm going to show you how to determine this on one of these antibiotic discs, and that is the erythromycin. So this is a zoomed in view of that one disc. So the E is for erythromycin. And what you need to do is measure the zone of inhibition. And so you're going to measure the diameter, so the whole width of this zone of inhibition. And you have to measure it using millimeters. So this is a ruler that has both the metric side, so millimeters, centimeters, and then the inside. You need to use the metric side. And so typically these numbers are going to be the centimeter measurements. So this one longer mark here is 1 centimeter. And so if you look, each of these individual small marks is 1 millimeter. So there are 10 millimeters in 1 centimeter. And so the goal is to measure this diameter. And so if you put your ruler up, you can see now I have the 0 lined up on one edge of the zone of inhibition. And then moving across, you can kind of see where the measurement is. And so again, the goal is to determine, is this bacteria, Staphylococcus, sensitive or resistant to this antibiotic? And so I'm going to measure the zone of inhibition. And so this line right here is about the measurement. So again, if each of these is 1 millimeter, that's 10 millimeters, that's 20 millimeters, that's 25 millimeters. And so this is 26 millimeters. That's measured, so 26 millimeters. And so once you have that measurement, there's a table that is provided for each of the antibiotics. And so you now have to determine, is 26 less than 13? Well, less than or equal to 13, which clearly it's not. Is it between 14 and 22, which it's not? Or does it fall in this category, which is greater than or equal to 23? So 26 is greater than 23, so we would conclude this bacteria is sensitive to this particular antibiotic. And hopefully this makes sense. The smaller the zone means the chemical is not as effective. So the bacteria are able to grow closer to where that disc is, closer to where that chemical is, which would indicate that chemical, that antibiotic, is not as effective. Whereas if it's a bigger zone, that means the bacteria grows much further away from the chemical disc, which would indicate the bacteria is sensitive to it. So that's how you would conclude your Kirby Bauer test. So if your plates have grown and you have confluent growth and you can see zones of inhibition, again, this is about 48 hours of incubation, then use the technique that I just showed you to measure the zones of inhibition and record that information in the data sheet that's part of the assignment. Additionally, you want to take photos of your plate. And this is the last lab experiment. And so if you've measured and taken pictures of this plate, we are done using all of the cultures. And so they all can be properly disinfected. So again, remember that is soaking all of the cultures in a 10% bleach solution for 24 hours. And then after that time, the bleach solution can be poured down the drain carefully, flushed with a great amount of water. And then any of those cultures can then be put in the trash.